Hello, dear students. In last lecture, we have discussed about the another sieve sampler is nothing but the slit type of air sampler. Now, in this lecture, we'll discuss about the most important air sampler is nothing but the Anderson air sampler. In this, the name itself suggests it is nothing but designed by the scientist Anderson. Uh, this Anderson sampler device is used selectively for the isolation, or we can say enumeration of microorganisms of different size of particle according to their size. So basically, this air sampler is also called a six-stage air sampler of Anderson. The sampler is an anti-tight cylinder that consists of a, or you can say anti-tap type of cylinder that consists of a stack of six metal sections. So already we have discussed. It is carried out in six different sections, which is made up from the metals. For each metal section, which is consist of perforated base, and the number of perforation is the same in each section. But depending upon the perforation for the differentiation between different particle size, the size of the perforation is progressively reduced from top of the column to the bottom. That means that air sampler is carried out in or prepared in six different metal sections. Each has a separate perforated section, and for each section we have to place a previously prepared sterile nutrient agar plate. In each perforated section, the number of perforations are same, but the size of the perforation, or we can say other words, the small opening or hole, is progressively reduces from top of the column to the we are moving progressively toward the bottom. To use this sampler, open agar plates are placed between each metal sections. By using electric motor, we have to suck the air from first opening from the air sampler so that the air uh, can enter into the air sampler, which is made up from the metal. And depending upon the particle size, the large particle or the large dust particle get trapped on the first section. As we are sucking the water air from by using electric pump, the air is again passed from the first section to the second section to third section, and depending upon the perforation size, slowly the different their particles are getting differentiated, and depending upon their size, they get trapped on the nutrient agar plate, which is placed individually in each metal section. And as we are moving downwards, the small particles are get trapped on the lowest metal section. In this, the viable organisms are collected or trapped on our plate, and similarly, we have to carry out the incubation of these plates so that we can get a proper count of different organisms which are present uh, on the different section. This diagram, it is nothing but the Anderson air sampler, which can be used for the air sampling. So it is nothing but the we can say it is nothing but prepared from the metal. Here is nothing but the opening of the air sampler, and that air sampler it simulate like the human respiratory tract, and depending upon the particle size, they get trapped on the particular each different section. So as we have discussed earlier, the opening at each perforation is similar for each section, but as we are moving downwards, the size of the perforation get reduced with respect to the particle size. So here, when a get entry into that particular metal section, the larger molecules get trapped on the first section. As we are sucking the air by using electric pump, the more air again pass from that particular first section to the second section. As depending on the particle size, they get trapped on the nutrient agar plate, which is placed already on that different sections, and they get trap, and the remaining air we have to remove from the air sampler. So what happened? This look like similar to that of the human respiratory system, and as we know, with respect to that, different particle size are uh, trapped at particular different location. Here, like first section is nothing but approximately particle size. It is greater than 7 micrometer, and it resemble with nothing but the nose or throat of the respiratory system of the human. Second section is nothing but the whose approximate particle size is 4.7 to 7 micrometer. So large particle getting trapped on the first section, that is nothing but the entry of your respiratory tract, there is nothing but the nose. After that, upper airway, then trachea or bronchi, primary bronchi, terminal bronchi, and after that, the small particle size having 0.65 to 1.1 micrometer size are getting entered into the alveoli. So this is nothing but the similar to that of the human respiratory tract, and by using that, we can use uh, or study the different microorganisms, or we use this different air sampler for the enumerating the different microbial count in the air. So depending upon the surrounding environment, we can use that particular sampler for different aspects. Thank you.